Hello everyone, my name's Jane Roberts, a Medical Affairs Dietitian at VitaFlow International Limited, and I'm very happy to welcome you to today's webinar hosted by VitaFlow titled Food as Medicine. This webinar aims to support healthcare professional education with regards to the dietary management of paediatric kidney disease. A few housekeeping items before we get started. The webinar is intended for healthcare professionals only. The webinar will last approximately 90 minutes and will include time for questions at the end. If you would like to ask a question, please use the question feature on your GoToWebinar toolbar. All questions will be screened and a select number will be answered by the speaker at the end of the presentation. The webinar is being recorded and will be available on VitaFlow's website for healthcare professionals in a few weeks' time. You can access PDFs of the presentation slides, a certificate of attendance, and a self-reflection tool in the handout section of the toolbar. The views expressed herein are those of the presenter and do not necessarily represent VitaFlow's views. The material is accurate as of the date it was presented and is for educational purposes only and is not intended as a substitute for medical advice. The learning outcomes for the session are documented on this slide. And now to introduce our speaker. Peter Stenvinkel is a professor and senior lecturer at the Department of Renal Medicine, Karolinska University Hospital in Sweden. He has published over 620 original publications and reviews, and over 30 book chapters on various aspects of inflammation, wasting and metabolism in chronic kidney disease patients. He is an associate editor of Nephrology Dialysis Transplantation and a fellow of the European Renal Association, European Dialysis and Transplant Association, and the American Society of Nephrology. Our chair today is Professor Rukhsana Shroff. Professor Shroff is a consultant paediatric nephrologist at Great Ormond Street in the UK. Professor Shroff has published over 300 original articles, reviews, and editorials. Professor Shroff works on international guideline committees for KDOQO, NICE, and ESPN. VitaFlow would like to thank Peter and Rukhsana for taking the time to take part in this webinar today. And without further delay, I'd like to hand you over to Peter.
Let's see how I can. There. So thank you, Jane, and it's great to be here. I'm really happy to be invited by VitaFlow to give this lecture about food as medicine. And we live on a hungry planet, and I think these pictures here that will be coming now, depicting what an ordinary family eats during 1 week in different parts of the world is extremely interesting. And for example, if you look in the top left corner, you will see what an US family eats during 1 week, and you can compare this to the lower right family, and peasant family from Ecuador, and what they are eating during 1 week. It is almost like they are living on different planets. And this will be one of the major problems for the next years. How should we feed the world and also sustain the planet? Now, when the population of our planet is more than 8 billion people, this is really something that we need to work with. This paper that was published in Lancet 2019 was an eye-opener for me. It's the Global Burden of Disease study, and they evaluated the health effects of dietary risks in almost 200 countries. And they could show that 22% of all deaths on this planet is attributed to dietary factors. And here you will see the list of the number of deaths at the global level attributable to dietary factors. And not surprisingly, on the top we have diet high in sodium, followed by diet low in whole grains, diet low in fruits, diet low in nuts and seeds and vegetables. And in fact, this study showed that suboptimal diet is responsible for more deaths than any other risks globally, and that include tobacco smoking. And I think you all agree with me that there is an urgent need for improving human diet across the nations on this planet. So usually nutrition science was not very complex. We dealt with undernutrition, micronutrient deficiencies, and metabolic diseases. But on this rapidly changing planet, the level of nutritional issues, the complexity of the human nutritional issues have increased markedly. And now we also may take into consideration climate change. We need to take into consideration nutrient changes in the crops that we are eating. Water scarcity is an emerging problem that we need to address. Land degradation, wildlife decline, loss of biodiversity. In that sense, especially the decline in pollinators on our planet is a major problem that could affect nutritional possibilities to feed our planet. Fishery collapse, we have pollution, and also displacement of populations. So this is a very complex scenario now. And as you're interested in nutrition, you're all aware of that food is just a bunch of molecules. Here we have some examples of the molecular structure of, for example, resveratrol in grapes, the sulforaphane in broccoli, the genistein in soybeans, the curcumin in turmeric. So actually, when we are doing cooking in our kitchen, this is almost like advanced chemistry lessons when we mix all these molecules. And I also like to introduce to you a new discipline. It's called the foodome. And this is a discipline that studies the food and nutrition domains through the application and integration of advanced omics technologies. And the reason for this is to improve the well-being, health, and knowledge. And there are now more than 25,000 substances that make up this complex foodome that you will find in the human diet. And the idea with studying the foodome would be to link the diet to the individual eating patterns, the individual foodome, the individual genome, and individual diseases. And this will then collectively being used as the foodome. And at the end of my talk, I will try to delineate more further how you could develop this concept for a more personalized, tailor-made nutritional approach. And this is just an example of studies of the foodome, and this is a study that was just published a couple of weeks ago. I find it very interesting, looking at the plasma metabolite profiles and the source of meat and fish consumption and the risk of type 2 diabetes. And here they could show that scores based on the identified metabolites for total meat, red meat, and processed red meat was significantly associated with the risk for type 2 diabetes. And it's these kinds of studies that I think will move the science and our knowledge forward. And this is what we should base our recommendations for nutritional treatments in our patients. So we have a number of burden of lifestyle diseases that we know accumulate as we get older. I have depicted some of them here, and you're all aware of the link between obesity, chronic kidney disease, Alzheimer, heart failure, cancer, osteoporosis, fatty liver, depression, and aging. And although all these diseases seems to be quite disparate, they have two interesting things in common. The first one being this chronic, smoldering, sterile, low-grade inflammatory response. And this is something that has been termed inflammaging. It's a combination of the words inflammation and aging. And also, these diseases have another thing in common, and that is changes in the gut microbiotic composition. Gut dysbiosis strongly linked to low-grade inflammation and all these diseases that accumulate as we get older. And in my talk, I will briefly mention a transcription factor called NRF2. I will show you soon more in detail about this, but I find it really interesting that irrespective of what disease, chronic disease that you study that are in this disease zone of aging, burden of lifestyle diseases, you will find low expression of this transcription factor. And I actually used 1 week of my time a couple of years ago to go through the literature, and it was clear that irrespective if it is cancer, diabetes, if it is Alzheimer, chronic kidney disease, obesity, or heart failure, the expression of NRF2 is usually low. And this is part of an intermediate inflammatory phenotype. And I would believe that irrespective of what burden of lifestyle diseases you are discussing, this is part of a phenotype that drives disease progression. We have here NRF2 depletion, we have loss of gut biodiversity, we have mitochondrial dysfunction, and we have oxidative stress. And we know that there are a number of established risk factors that promote this intermediate inflammatory phenotype. We know that drugs, we know that sedentary lifestyle, we know that poor nutrition, smoking, infections, all drive this intermediate inflammatory phenotype. So if you ask me how should it enable better aging, I would guess that this is the phenotype, the intermediate inflammatory phenotype is what we should try to target. And it's actually possible to target this phenotype not only by pharmacological means, but also with bioactive nutrients. So you remember this family from Ecuador on the top here, and the family from USA. And you can see that they have a completely different dishes on their table. And I'm sure you can all agree with me that these two different styles of eating will have very different effects on the foodome. And this actually serves as a basis for a paper we published in Nature Reviews Nephrology a couple of years ago, evaluating food as medicine and discussing the potential benefits in the context of chronic kidney disease using this approach. And I find this extremely interesting concept of targeting the uremic phenotype by food as medicine. We have many opportunities here. We could target transcription factors involved in inflammation and oxidative stress. And here we have this NRF2 that I will discuss very soon. You could target these transcription factors with bioactive nutrients such as sulforaphane that you find in broccoli, curcumin you'll find in turmeric, anthocyanins in berries, flavonoids in red fruits, quercetin in apple, cinnamaldehyde in cinnamons, and rhizome in ginger. And of course, there are many other examples that I could not list here. And NRF2 is a two-component system, and it's composed of NRF2 and its inhibitor KEAP1. And this is like a molecular switch, that if you turn this molecular switch on, there will be an increased expression of hundreds of cytoprotective genes that will increase the clearance of oxidized and damaged proteins that will reduce oxidative stress and inflammation. And this transcription factor is something that you can actually affect by your living habits. Studies show that psychosocial stress, uremic toxins from Western diet, and smoking will all downregulate NRF2. Exercise is something that will stimulate this transcription factor, and then we also have a number of synthetic NRF2 stimulators that we can use as drugs. The most well-known of these is bardoxolone, but we also have other drugs that we already use in the clinic that stimulate NRF2, such as metformin, statins, and lithium. But for the concept of this talk, we should be more interested in the bioactive compounds and fermented food that can also stimulate this transcription factor. Curcumin, rosemary, tomato, and coffee are all examples of bioactive compounds. Kimchi, the Korean fermented food usually served as breakfast, is something that also stimulates NRF2. But the most potent NRF2 stimulator found in broccoli sprouts, and this is a stimulator called sulforaphane. And sulforaphane has well-documented effects on NRF2 and inflammation. Sulforaphane modifies the tile groups of KEAP1 and thus increases the availability of NRF2 to the nuclei. It binds to essential cis residues and inactivate NF-kappa B, and it also reduces the activation of the NLRP3 inflammasome. And sometimes a picture says more than 1,000 words, and I think this is a very excellent example of the properties of sulforaphane, comparing two different mice, aged mice, one of which received the control diet on the left, and then we have the similar mice that receive sulforaphane, and it's quite clear that those that receive sulforaphane actually have a much healthier and younger appearance. Here you will see what I think is to date the best clinical study evaluating the effect of broccoli sprouts in patients, and these are patients with type 2 diabetes. First, they could show that sulforaphane in increasing concentrations actually stimulate the nuclear NRF2 effects. But the more interesting parts are actually the clinical parts, and they could show in type 2 diabetics that treatment with broccoli sprout extracts decreased fasting blood glucose levels, the glycated hemoglobin levels, and they could also show here, evaluating the glucose production from the liver, that sulforaphane improves glucose tolerance by a magnitude similar to that of metformin. So broccoli sprout extracts seems to have clinical beneficial effects, and we are currently finishing a randomized trial here in Sweden evaluating the effects of broccoli sprout extracts in patients with chronic kidney disease stage 4. We have now achieved 99 patients, and we are targeting for 100, so there's only one more to go. It is also possible to use food as medicine to target mitochondrial biogenesis. You could use it to target nitric oxides, and here is something that you can do with foods like radish, beetroot, and spinach. And when you ingest something that is rich in nitrates, such as beetroots, Nitrite will in the oral cavity through effects of facultative anaerobic bacteria be metabolized to nitrite. Further, after passing the gastric part, it will be metabolized to nitric oxide and further on to citrulline and arginine, which we know have beneficial effects on endothelial cells. I found it so interesting when evaluating the literature here that the oral cavity and its bacterial composition is something that we not really take much into consideration, but there are numerous factors that will affect the oral cavity and thus the synthesis of nitric oxide. In the green box, you will find that smoking, periodontal disease, proton pump inhibitors such as omeprazole are actually downregulated nitric oxide synthesis. And also very interesting to find out is strong link between the use of antibacterial mouthwash and NO synthesis. And there are actually quite a large number of studies showing a strong link between the use of mouthwash and hypertension and poor response to antihypertensive drugs. So now after finding out of this, I always ask my patients that have a poor response to antihypertensive drugs if they are using mouthwash or not. So in this toxic uremic milieu, we have like a perfect storm that contribute to premature aging phenotype. We have oxidative stress, we have signs of DNA damage, telomere shortening, we have senescence with accumulation of P16, mitochondrial dysfunction, and accumulation of uremic toxins. These are a milieu that will promote the appearance of senescent cells. And senescent cells, which I will discuss more in detail soon, will, via a number of regulators, stimulate the premature aging phenotype. And in this review, we discuss how our nutrients could affect these senescent cells. And let's start first with the effects of red meat. And talking about red meat, we need to discuss TMAO and the surprising link between the gut and the heart health. So TMAO is produced from food rich in L-carnitine, and we know that red meat is particularly high in carnitine. A recent paper just published a couple of weeks ago in Nature Microbiology showed that when you eat red meat, this will shift the gut microbiome to produce more of the precursor of TMAO, that is TMA, and this is done via microbial gene cluster. So every time you eat red meat, your gene cluster in the microbial part of the gut will determine how much TMAO will be produced. And on top of this, it's also been shown that red meat reduces the ability of kidneys to excrete TMAO. So patients with kidney disease, they have markedly elevated TMAO levels. And this has been hypothesized to be the new red meat risk factor. And the literature is now inundated with reports like this one showing that TMAO induce an aging-associated phenotype. This is another recent study showing strong associations of TMAO with stroke risk in the general population. Studies like this one show relation between TMAO and hip fracture in postmenopausal women. And also TMAO is something that is a prognostic marker for incident cardiovascular events beyond traditional risk factors in acute coronary syndromes. So taken together, high TMAO seems to predict all these burden of lifestyle diseases that accumulate as we get older. And the link between TMAO production in the liver from TMA is very interesting and related to what we eat. If we start with red meat, this is a rich source of carnitine, and carnitine will be metabolized by the gut microbiota to TMAA and then metabolized to TMAO in the liver. And there is no doubt that when you eat a meal with red meat, as depicted here on the right, you will find significant increase in TMAO levels comparing to white meat or non-meat meals. Then we have eggs, and eggs is a source of phosphatidylcholine and choline, and in theory this could also generate TMA in the gut. But now there's a study that evaluated the effects of eggs and choline supplements on fasting TMAO levels in participants with normal renal function. And this slide here and this figure here show that in 18 controls, the ingestion of 4 eggs per day during 28 days did not lead to any increase in TMAO levels. In contrast, choline supplements increased TMAO. So this is good news for all of you that like to eat eggs. And then we have the most interesting part of all, and that is the fish TMAO paradox, because fish could be a rich source of TMAO, both TMA and TMAO. And this is kind of puzzling, because generally we regard fish consumption as something that could benefit your health. To understand this paradox, we must learn and know the reasons why we have TMAO in nature. And TMAO is an osmolyte that counteracts the effects of different destabilizers, such as low temperature, high urea, and high hydrostatic pressure. So animals that are are exposed to low temperature, high urea, and high hydrostatic pressures, they should have extremely high TMAO levels. And we have animals that, for example, living in very deep water, such as this Notoleparus kermadescensis, which is the second deepest fish ever seen alive, that could help us a lot to understand this. And then a recent study in PNAS could actually relate the depth of where the fish was caught and the TMAO levels in their muscles. So the deeper these fishes are living, the higher the TMAO. So there is a higher muscular TMAO content in fish habitating deep sea. And another recent study, we could learn even more about TMAO. And here they have looked at the concentrations of TMAO and TMA, that are the red bars, with omega-3 fatty acids, the eicosapentaenoic acids, the blue bars, and you can see that there is a tremendous difference in the concentrations. And we have some fishes and seafood that there is actually no measurable TMAO at all. These are the fishes and seafood to the left part of this figure, including, for example, mussels and trout, especially rich in EPA, but there is no TMAO. And on the other hand, you have deep sea fishes and seafood such as lobster and cod, which would have very high levels of TMAO and low levels of omega-3 fatty acids. And when healthy subjects were given a meal with different seafood, you can see the tremendous difference in TMAO levels. If they were given a meal of shrimp or canned tuna, there was no change in TMAO. But if they were giving a meal with wild salmon and especially fish sticks, you could see an enormous rise in TMAO levels. These are healthy controls, so one day after the meal, the TMAO levels have returned to normal, but you can guess that in patients with reduced renal function, TMAO levels may start to build up. And especially worrisome, I think, is this tremendous increase in TMAO levels following intake of fish sticks, because this is one major component of the food that we serve our children in the school here in Sweden. So with this in mind, we just published a study here conducted in more than 800 patients, older patients with incident CKD stage 4. And these were part of the EQUAL cohort. They were followed for 48 months. We measured trimethylamine oxide, TMAO, and this we regarded as a reflection of red meat, and then also carboxymethylpropylfuran CMPF, which is a metabolite of furan fatty acids, and it's a reflection of the intake of fish. And then we just made a very simple division into 4 groups based on the median levels. So we had one group here that have high TMAO but low CMPF, and this is a group that is suggesting to have a high dietary intake of red meat but a low intake of fish. And we were quite surprised when we evaluate these patients after 4 years, because it was obvious that especially this group have a higher all-cause mortality and also much higher incidence of starting kidney replacement therapy. So if these data can be confirmed, I think we really need to tell our patients with chronic kidney disease that they should avoid especially processed red meat and instead try to get their protein sources from fish. And then probably, based on the results from the other study here, we should advise them to avoid fish from deep sea. And there is no doubt that eating too much red meat may speed up the aging processes. This is a newsflash from a study I was involved in a couple of years ago. We evaluated men in Glasgow's poorest suburb. It's a name called Carlton, and here the average man will only become 54 years old. And then we compare the results to inhabitants, males in the suburb of Lenzie, just a couple of 15 minutes ride from each other. And in Lenzie, the average male will become 82 years old. So there's a difference in the male life expectancy of almost 30 years. And we can really see that there was a huge increase in cheap processed red meat consumption in this suburb with where the males only became 54 years old. But we also have more robust data, such as this recent Nature Communication paper showing that genes involved in metabolizing iron were associated with a healthier, longer life. And these findings can then explain why high dietary levels of the iron-rich red meat has been linked to many age-related conditions. Not only cardiovascular disease, but also cancer disease, such as colon cancer. And then we should not forget that this transcription factor, NRF2, is actually something that orchestrates the activation of antioxidant defenses that are so crucial to protect us against iron toxicity. So in conditions where we have an inflammation and low NRF2 expression, it may be that iron is more toxic. So that was a discussion about red meat, but we also have bioactive nutrients that could actually have beneficial effects on the senescent cells and actually inhibit the activities, the effects of senescent cells, and decrease the number of senescent cells and the inflammatory effects. And this leads me to these senescent cells. And senescent cells are extremely interesting. And as we get older, senescent cells start to accumulate in our bodies, whether we want it or not. Usually the immune system can clear the senescent cells, but when we get older, and especially if you have chronic diseases, they accumulate. And the senescent cells are like zombie cells. They are non-living cells that makes a lot of trouble. And the main trouble is that they actually poison the tissues with pro-inflammatory products. This is what's called the SASP response, the senescence associatory secretory phenotype. So eventually the immune system failed to kill the senescent cells that start to grow in numbers and also move to other parts of the body. So the emerging concept would be to find drugs that could actually make senescent cells go into apoptosis. And senescent cells is thus an emerging target for all diseases of aging. And we have a number of senolytics or xenomorphics in the nutrients. Dasatinib, quercetin, and fizidins are maybe the best known of these senolytics. They reduce senescent cell and bondage without much of the off-target effects. The idea here will be to test natural plants that possess a high concentration of these senolytics, give it to old mouse with senescent cells, and then to see if these senolytics delay aging, reduce the age-related diseases, extend health span, and then we'll have less senescent cells. Next step will be to conduct clinical trials, and such trials are currently ongoing in order to develop more personalized precision nutrition. And there are studies like this one now showing that in aged or atherosclerotic mice, chronic senolytic treatment alleviates the vasomotor dysfunction. This Nature paper is even more interesting, showing that intermittent oral administration of senolytics, that is dasatinib and quercetin, actually alleviate physical dysfunction and increased post-treatment survival by 36%, and also reducing mortality hazard to 65% in mice. So if this can be reproduced also in patients, this is a tremendous opportunity to affect the age span in patients with chronic burden of lifestyle diseases. A couple of words about the microbiota, and you're all aware of the tremendous importance of an intact microbiota and the effects of disturbed gut dysbiosis and the leaky gut syndrome that emerges, the translocation of bacteria and bacterial products that activate systemic inflammation and promote premature aging. And there are so many different bacteria out there. This is a very simple example of the good bacteria, such as bifidobacteria, lactobacilla, and then we have, of course, a range of bad bacteria, Campylobacter, Enterococcus, and in chronic kidney disease, I think we have all been seen patients suffering from severe Clostridium difficile infection. And here we have an emerging problem that has not been much discussed in the renal community, but looking through the literature, you get really scared when you see that all these microplastics that emerge in humans and animals due to plastic products in our environment has huge impact on the gut microbiota, in impact that will drive this disease of aging with NRF2 depletion, mitochondrial dysfunction, oxidative stress, and loss of gut biodiversity. And I read somewhere, I don't recall where, that actually that each week we'll get as much plastic as you will find in one credit card. And during this industrialized period, we are now witnessing a change in the microbiotic composition. We know that the microbiota has a tremendous impact on the immune function, metabolic function, the nervous system. And actually, we are now witnessing something that we've never seen before in Homo sapiens, and this is this industrialized microbiota, a changing microbiota and loss of biodiversity. I was surprised to find out that air pollution is something that actually affects the composition of the gut microbiota. Studies in an animal, this is a lizard, shows that actually with 2 to 3 degrees warmer climates, this will cause a 34% loss of the diversity of the microbiota. And then we have, of course, the plastics that will affect the microbiota, both in our nose and in the intestine. But of course, the most major cause of change in microbiota is the processed food, or the ultra processed food, the key hallmark of the Western diet. And we know now that processed foods drives intestinal barrier permeability and the microvascular diseases. This is a study showing that ultra-processed diets cause an excess caloric intake and weight gain. We just get hungrier eating ultra-processed food. This is a study in Nature showing that artificial sweeteners induce glucose intolerance by altering the gut microbiota. And the problem is that this ultra-processed food is addictive. We want to eat more of it. It stimulates our dopamine system. And a recent study in JAMA Neurology is really scary, showing in Brazilian patients an association between the consumption of ultra-processed foods and the cognitive decline. And of course, ultra-processed food is also a major risk for incident chronic kidney disease. This is a prospective cohort study of almost 15,000 patients in US. The outcome was incident chronic kidney disease, and they evaluated quartiles of servings of ultra-processed food per day. The top contributors to ultra-processed food was, of course, the sugar-sweetened beverage and the ultra-processed meats. And they find that there was a 24% high risk of incident chronic kidney disease for the highest versus the lowest ultra-processed food consumption quartile. And for each additional serving of ultra-processed food, there was a 5% higher risk for chronic kidney disease. So this is from a recent review we just published. This ultra-processed food is a major problem we find in chronic kidney disease. It's really a double trouble, because this ultra-processed food will promote a number of comorbidities. It will cause metabolic changes, and on top of this, it will also stimulate a number of complications in chronic kidney disease, such as dysbiosis, hyperkalemia, metabolic acidosis, hyperphosphatemia, and insulin resistance. So going back to this figure of food as medicine, we have then the dysbiosis, which we already discussed a lot, and here we have a number of opportunities to affect this with fibers, prebiotics, soy food, polyphenols, and urolithin. Urolithin, I think, is extremely interesting, and this is a major microbial metabolite that you will find in polyphenolics of berries and pomegranate fruits. It has anti-inflammatory, antioxidative, and anti-aging activities. This Nature communication paper show that urolithin A actually exert barrier functions through activation of NRF2 dependent pathways, and it kind of upregulate the epithelial tight junction proteins. So the gut will be more tight, and there will be no leakage or less leakage of inflammatory products. So treatments with urolithin also was shown to attenuate colitis in the preclinical models, and this was done by remedying barrier dysfunction and having anti-inflammatory activities. So this really suggests that urolithin that you get from blueberries and other berries may actually be something that we should provide our patients with chronic kidney disease with. And there are, in fact, a number of quite good now randomized trials with blueberries. This is a paper in Circulation showing that high anthocyanin intake from blueberries was associated with reduced risk of myocardial infarction in young and middle-aged women. And here we have a 6-month double-blind randomized control trials with blueberries, and it was shown to improve many biomarkers of cardiometabolic function. And actually the authors concluded that with effective size predictive of 12 to 15% reductions in cardiovascular risk, blueberries should be included in dietary strategies to reduce individual and population cardiovascular disease risk. So let's go back to these inhabitants in Glasgow. Carlton, I already told you about, and we have then the Lenzie, the rich suburb, those with a high socioeconomic position. And here we studied the circulatory microbiotic differences within these two suburbs. And the blue bars represents those living in Lenzie, and the red bars those living in Carlton, the low socioeconomic position. And you can see there is a tremendous difference in the microbiota. And for example, the good bacteria such as lactobacillus are found for higher levels in high socioeconomic positions. So those living in high socioeconomic positions, they actually possess more circulatory beneficial microbiota. So the Times found out about this study, and you can read in June 22 that unfriendly gut bacteria is linked to early aging. I think here we have the main problem in this Carlton suburb is that the stores look like this. This is an example of a food desert. You have food, but it's mostly processed food, food with a lot of additives, food with a lot of phosphate, and there is really hard to get fresh vegetables and fruits in these stores. And also I should not forget to state that these salutogenic bacteria that you will find in these rich suburbs are the same bacteria that are having a capacity to metabolize and produce agonists of NRF2. So here is a recent Nature paper I found extremely interesting, because here they have found from 1 to 2,000 old people living in Mexico or Western USA, and they could actually get well-preserved DNA from these 8 old people. This is something they call paleophysis. And they could then find previously undescribed microbial species, and this could help us to elucidate the evolutionary history of the human microbiome. And this is just one of many interesting figures from this paper, showing that in these 1 to 2,000 year old people, there was a number of bacteria that actually was not seen in the human industrialized microbiota, for example, the Solobacterium and Prevotella. And Prevotella is a really interesting bacterial species, because when you eat a lot of fibers, you will get more Prevotella in your microbiome, and Prevotella is essential for the metabolism of carbohydrates. And this could be one link to the epidemics of diabetes and obesity in the industrialized society. So the microbiota has changed a lot from the hunting and gathering period to the ancient agriculture period and to the present modern industrialized period, where we have a lot of processed food and also drugs that will affect biodiversity and function of the microbiota. And maybe there is something that we have lost in the modern Western diet, and this is something discussed here, that we have a lot of foods that we are not really eating a lot these days, especially the fermented food that we know activate NRF2 cell defense pathways. And this leads me to the fermented food, which I think is a good opportunity to change the phenotype of patients with chronic kidney disease. We know that fermented food is something that stimulates NRF2, it inhibits NF-kappa B, it has effects on the gut microbiota. And the paper on the left is published in Cell, and this is, I think, a really good paper, a prospective randomized multi-omic study, and they investigated the effects of high fiber fermented food, and they could clearly show that both high fiber diet and fermented food increased the microbiome diversity and reduced inflammatory responses. So this could be a way to reintroduce evolutionary important foodborne microbes that we have lost over the course of sanitizing the industrialized environment. So the last part of my talk, I will go a little more into planetary health, and we as Homo sapiens, which actually means the modern man or the wise man, is responsible for this ongoing 6th major mass extinction. And to develop and control our environment, we have often taken shortcuts that are both selfish and short-sighted. We have failed to consider the long-term consequences. And now this unprecedented improvement in health that we have seen over the last 100 years, and that was of course before COVID, is actually now threatened by disturbances in the global environment, the loss of biodiversity, the global warming, the pollution, and this disorganization and climate change. And during the current period, which is actually called the Anthropocene, you will find that things are out of control. And these are just figures showing the massive increase in carbon dioxide, stratospheric ozone, the marine fish capture, ocean acidification, methane, domesticated land, etc. And WHO has calculated that 23% of all global deaths today are more or less linked to changes in the environment. So this is a major health problem, and it will become bigger. And here I would like to come back to this intermediate inflammatory phenotype that we discussed, that promote a number of burden of lifestyle diseases that accumulate with age, because literature is now showing that all these environmental stressors, such as global warming, greenhouse gas emission, air pollution, pesticides, heavy metals, stimulate this very same intermediate inflammatory phenotype. And this may be the reason why the changes in environment will increase the risk for all these burden of lifestyle diseases. But it may also provide opportunities, because treatment opportunities that manipulate the exposome for the prevention of aging and environmental stress may overlap. So this is from a recent paper I just published together with Paul Shields and Rick Johnson, discussing the Anthropocene, the current period when humans see themselves as holders of the environment and natural resources. Clearly, we humans are not well synchronized with the rest of the planet at the moment. Actually, today, the human-made global mass surpass all living biomass. Just start to think about this. This is just extremely annoying. And during Anthropocene, we have an organizational structure that actually has shown that the natural resources have been mostly used to serve human demands. And this is just a word cloud depicting a number of factors that characterize this current Anthropocenic organizational structure. Humans protect themselves against nature. There is a massive deforestation, defaunation. We disregard lessons from previous mass extinctions. There is a monoculture farming, monodisciplinary research in silos, loss of biodiversity, pro-inflammatory lifestyle, etc. And I believe that central to any actions to aid the planet now is to first recognize that the current crisis we are witnessing actually require that we move away from this anthropocentric perception. And this is something we really need to convince our politicians to do. And the Eat Lancet Commission published this paper in 2019, and this may be one of the most important papers ever published. It showed that food is the single strongest lever to optimize human health and environmental sustainability. And the environmental impacts of food productions are enormous. It's actually responsible for 27% of all the greenhouse gas emissions. 70% of all the fresh water use on our planet goes to food production. It's the main driver of biodiversity loss and tropical deforestation, and it also is something that affects the risk for future pandemics. And here is a list of how much water it takes to produce the different food sources, and you can see that tomatoes, there is not too much water, although 214 liters per 1 kilo tomato sounds a lot. Avocados have usually been blamed for using a lot of water. It is almost 2,000 liters per kilo, but compare this to the production of 1 kilogram of beef, 15,400 liters, and then you have the peeled almonds, which is on the top of this list. And the emission gap from food consumption is something we really need to do something about. And you can see that today the emission gap from eating is around 5.6 gigaton of carbon dioxide. And if we continue to eat as we do today on the planet with a growing population, by 2050, this will be almost doubled to 9.8 gigaton. But to accord to the Paris Agreement, we really need to change our eating habits to decrease the emission gap from food consumption to 5 gigaton. And there are some important facts about food production, which I think will be interesting for you to know about. And maybe this you're all aware of that 30% of the food production is never eaten. And this is just extremely annoying. 70% of the soy produced on our planet are used to feed animals. 36% of the calories produced in the world are used to feed crops into animal products. And 70% of the world's food is produced from only 12 plants and 5 animal species. So this is not an example of good biodiversity. And I think this is maybe one of the major problems today, this monocultural farming. And I found it interesting. Now it's been a lot of media attention to Queen Elizabeth in the UK. I think she was a feisty woman in many ways, and I was really glad to learn that when they interviewed her former royal chef, that Queen Elizabeth refused to eat out of season fruit in winter. And in the interview, he informed us that when there was an out of season fruit or vegetable, she will return the plate and say that they have to do better. And probably this is what we all should do. We should not eat fruits and vegetables that have traveled around the world to enter to our supermarkets. This recent PNAS study was, in my opinion, really an alarm bell. That I was not aware of this, that agriculture is actually a major contributor to air pollution and is the largest environmental risk factor of mortality. And in this paper, they could show that dietary shifts toward more plant-based foods that maintain protein intake and other nutritional needs could actually reduce the agriculture air quality related mortality by over 60%. So these are major changes. And we have kind of a luck here, because it seems like the unhealthiest food is the food that also have the highest environmental impact. This is another PNAS paper. They evaluated the health and environmental effects of different foods. And you can see here the green dots are foods that are good both for the health and environment, but then the red dots, these are those that are neither good for health and environment, and here you will find unprocessed and processed red meat. And dietary transitions toward greater consumption of healthier foods is also something that could improve environmental sustainability. And also we should then recall that carnivorous diet is something that drives the disease of aging. And here, if we link the risk of mortality on the x-axis to the average relative environmental impact, you will clearly see that unprocessed and processed red meat are outliers in the top right corner, whereas green foods are those with the low relative risk of mortality and the low risk of environmental impacts, are foods that will actually should consume more of. And the global meat production have increased markedly from 61. You will see this tremendous rise, and this is specifically attributed to an increase in the meat production, especially from pork in China. If you look at the per capita meat consumption 2019, Portugal is on top of the list, but then you can see that the major part of the Portugal meat consumption is actually from fish and seafood. If you go to look at beef, you will find that the main consumer of beef on our planet is now Argentina, 48 kilogram per person a year. And this is another slide evaluating changes in global temperature. That is the line, and then the increase in the global meat production. And here you will find an excellent correlation. Of course, we don't know what the cause and effect is here, but there is nevertheless a strong association. So the more red meat we eat on our planet, the higher the temperature. And when you look at meat, I think it is extremely interesting to compare the environmental effects, for example, for soy. And here you can see that with the same protein content, the red meat will have a major increase in the land use, a major increase in the amount of water used, and also a major increase in the use of fossil energy. So if you look at the nutritional efficiency of soy, if you have 9 kilograms of plant protein, you could actually serve 68 portions of tofu. If you instead give 9 kilograms of plant protein to a pig, this could give you 12 portions of pork meat. And if you gave 9 kilograms of plant protein to a cow, this will result in 5 portions of red meat. And I would say that the figures here that is shown is quite scandalous, that of the global food production, only 20% are directly given to humans to feed them, and 76% of the global soy production go to feed the animals, especially poultry and pig. So we are currently in a lose-lose quadrant, because the food we eat is neither good for our health or the environment. And I already told you about foods that we should emphasize, including vegetables, fruits, legumes, whole grains, and nuts. And there is an extreme imbalance in the global eating habits. And here you will see that especially the red meat and the starchy vegetables is something we eat way too much of, maybe also eggs, but we, on the other hand, eat way too little of the fruits, vegetables, and the legumes. So what we need to do now is to try to move from the lose-lose quadrant to a win-win quadrant. That is that the food we eat is both good for the environment and the health. And in EU, meat and dairy cause 83% of all the greenhouse gas emissions. And here you can see that meat provide 56% of the gas emissions and dairy 26%. And I think the main problem is this, because the plant proteins that we grow on our planet is now going to a little part of it is going, about 20% is going to feed the human population, whereas the majority is now used to feed animals such as pigs and cows. And then we eat the red meat from these animals. And this is something that will increase the greenhouse gas emission, and it will also stimulate the disease of aging. So this is clearly nothing that will work in the long run. We need to change this and drastically reduce the amount of plant proteins that go to feed animals. This will lead automatically to a much lower consumption of red meat, and then we should use the major bulk of the plant proteins to feed the increasing population on our planet. So we should be talking about prioritizing food for people rather than feeding for the excessive numbers of farm animals exploited for food production. And you remember this, one of my first slides about food is just a bunch of molecules, and this is, I think, something we could use by this new discipline, the foodome. And if you could take into consideration for all of us our age and sex, if we could take into consideration the differences in the genome, our individual food preferences, what kind of non-communicable chronic diseases we have, our metabolome, the current medication, and the gut microbiome, and if we could then also exclude all food items that are not sustainable, use artificial intelligence to make this into our own individual QR code. And of course, during our lifetime, this code will change. We get older and there will be differences in the food preferences and our metabolome and the medication. So this QR code is a moving target that will change. And wouldn't it be nice if you can use this QR code when you go to the supermarket, and when you go into the supermarket, you show your current QR code, you will get a list of food items that will be good for your health and for the environment. This will be linked also to some recipes, and you can walk out from the supermarket with your own individualized grocery bag. And maybe even you have your same QR code when you go to a restaurant, and you will give it to the personnel there, and then they will make this into a good meal for you. So my main message is that radical transformation of the global food system is urgently needed. And of course, this needs to be based on solid scientific support. But if we could change to locally produced plant-based diets, grains, fermented nutrients, and limit the intake of red meat, this could have enormous beneficial effects. First, it will reduce the risk of burden of lifestyle diseases, no doubt about that. It will reduce the greenhouse gas emission on our planet, no doubt about that. But also, I think this kind of diet could increase our resistance against the huge environmental stressors that we are now fighting on our planet. So this is a win-win-win situation, and I think this is not very common to have, and I think it would be extremely interesting if this could radical transformation could be implemented and tested. So I have, during my talk, tried here to connect some dots, and the central part has been this intermediate inflammatory phenotype that we know is so strongly associated with aging. I also told you that the current climate crisis with air pollution, global warming, and pesticides and warming will stimulate the same intermediate inflammatory phenotype, and the result of this will be an increased risk for non-communicable chronic diseases. Somatic mutations play a central role here. But I also told you about some possibilities, or maybe one of the possibilities I did not have time to talk about, the biomimetic lessons from the animal kingdom, because there are a lot of ingenious solutions developed in the animal kingdom that could help us to solve this problem. But maybe I can talk about this another time. But what I have talked about is this possibility to use food for health to interrupt this intermediate inflammatory phenotype. So the power is on your plate. And I think this is something we must and we can do to change our eating habits, not only for the sake of our health, but also for the sake of the environment. And with this, I'd like to thank all my collaborators in different aspects. I'm working mostly on early vascular aging, but also with nutritionists on food as medicine and biomimetics, which is a really thrilling and interesting research area. Thank you.
Thank you very much, Peter. That was absolutely fascinating. And it is really wonderful to see how much good science there is behind what we were told is this is good for you, so eat it. I'm delighted to see it backed by so much excellent science. May I encourage people to send us questions in the question and answer box you have on your screens? And to start, perhaps, Peter, may I ask you a couple of questions, please? From this really interesting talk. So you talked about a very interesting association between ultra processed foods and CKD progression. May I ask, is this like a vicious cycle? So the worse the CKD, the less excretion of some of these toxic products, and the worse the condition gets?
Yeah, absolutely a vicious circle. I think the chronic kidney disease progression is just an excellent example of how vicious circles start. So, and then the accumulation of uremic toxins will make this even worse. So that's why chronic kidney disease and ultra-processed food don't go well together. This is really double trouble. And it's, I think, something we really need to talk to our patients about, that they should avoid this. The problem is that ultra-processed food is easy to get, it's quite cheap, and it's good. You want more of it, so it's not an easy task to convince our patients to change the diet, but it's extremely important, I think, and of course you should try to start as early as possible.
Thank you. And as you showed so nicely from the example from Glasgow, it's sadly the less the poorer parts of society who are victims of this more than anyone else, and then it just feeds into that vicious cycle. Peter, what about reversibility? So if somebody were to try and change their ways and go from a largely carnivorous diet to a largely vegan diet with all of the healthy food, are there any studies to back up and show that there can be reversibility?
Well, we are actually conducting one of those studies now. We are providing patients with CKD stage 4 and 5 healthy food bags that has been developed by our nutritionist. So we provide them these food bags for free for a couple of weeks. And then we study if the main objective here is, of course, to evaluate potassium, so that there will be no major increases in potassium. But then we see how the impact on their quality of life and risk factor profile, uremic toxins, etc. We have not completed the study yet, but the patients that have been in the study, they are very well. I've never seen so motivated and happy patients, because we provide the food bag for the whole family. And the majority of them will want to continue with this healthy plant-based diet and reduce meat intake. And then also they have been very happy because this was an opportunity for them to interact with a dietitian on a regular basis. Every week they could talk to a dietitian. So that really made a difference for many of them.
That's fantastic. Excellent. And on that note, which is so interesting, if a CKD patient moves towards a largely plant-based diet, potassium can become a problem. Now we routinely use phosphate binders without worrying about it. Do you see a time where we'll routinely use potassium binders and then encourage a high fiber, high plant-based diet?
Yes, this is what we are doing now. So potassium will increase when they change the diet, but it is possible to control it with. We use Lokelma in this study, and it's well controlled.
Lokelma.
Yeah.
Excellent. Thank you. And there are people who like to eat whatever they want and then take their statins and take their medicines and feel not too bad about it. Have you ever considered, or will there ever be a study of a strict, good, healthy diet versus taking a tablet of bardoxolone, for example? I know it's an extreme example. I understand.
Yes. So as I mentioned, we are currently actually just finishing this study of broccoli sprout extract to see if that will have an impact, and that's a randomized trial. If this will have a beneficial effect on the risk factor profile, this could be really an incentive to start larger studies with targeted nutritional interventions. And but it is extremely complicated to do these studies because we all have our individual microbiome. We have different genomes. The metabolome is different. So there are so many factors that need to be taken into consideration. But studies like, I think the one I showed on how the metabolome after red meat ingestion, how it increases risk for type 2 diabetes, these kind of studies will really move this field forward a lot.
Thank you. And there's an interesting question from the audience that you talked about the TMAO levels and TMAO being in carnitine or produced from carnitine. Now, we actually give intravenous carnitine to some patients, or even oral carnitine supplements, thinking that it's actually good for myocardial function. And equally, I believe young athletes may be taking it. Would you have a view on that?
Well, the carnitine could be a double-edged sword, but the carnitine I'm talking about is not the infused one. It's the carnitine that you will find in red meat. And of course, red meat has many disadvantages, but we should not forget that red meat is also an excellent source of high quality proteins. In some patients, they may need iron. So I really believe that the consumption of red meat need to be adapted to sex, because there are usually males eat much more red meat than females. And the age, and there may be different periods of life, maybe during teenagers that grow a lot, menstruating women need iron, and so maybe we need to change and adapt our red meat intake to both sex and age. And I have a feeling that talking to my patients, especially middle-aged males, they are more carnivores than the females, and many of my patients, they think they are lions. They can eat red meat on a daily basis, but I try to tell them that they are more like monkeys. Our digestive system is more like the primates than the lions, and we are not designed to eat red meat on a huge and daily basis.
That's fascinating. So I was actually going to ask you and link this question up with your extremely interesting work where you look at, you find lessons from the animal kingdom that can help improve our health and well-being. With the so what is it adapted in animals that allows them to have a largely or entirely carnivorous diet? Or even the examples with hibernation you give and inflammation, etc.
Yeah, well, the problem in for example, felids that are obligate carnivores, they must eat red meat because they need it for the metabolism, and they cannot do without red meat. But now we see the consequences with a really high risk for chronic kidney disease. We know domestic cats, 1 out of 3 domestic cats that are old, they will die of chronic kidney disease. And now when I'm collaborating with veterinarians and zoologists, I also learn that tigers and lions are very susceptible to chronic kidney disease. When they looked in an autopsy material from German zoos, they find that almost 90% of big cats have severe renal lesions. And when you measure creatinine in a tiger, you will find that they have levels comparable to what you find in patients with CKD stage 3. And
wow.
So we are currently conducting a study when tigers are fed red meat, then we switch to white meat, and then to fish, and we will see what impact that will have on the renal parameters.
Will you test the tiger's quality of life as well as you put them on a strict diet?
Yeah. But we
I
need
hope they're
a few
happy.
volunteers to feed them.
Yeah, I'm sure. I'm sure. And there's a question here from somebody who asked, can you please explain what an SFN diet is?
Sulforaphane. That was the broccoli sprout extract.
Yes, yeah.
Okay.
Thank you.
So
And
sulforaphane, you will find it in broccoli, but then you need to eat enormous amount of broccoli. The sulforaphane is especially found in the sprouts. So that makes it a little more difficult to get this. You can grow your sprouts yourself, and there are ways you can also buy it. But broccoli is good for other reasons, but it's not much sulforaphane in broccoli per se.
Right. Okay. Thank you. And that was another question: how much broccoli would I need to eat to benefit from it? Are there
many kilograms of broccoli? But broccoli sprouts, we have a powder that you can just put on your porridge or what you eat for morning, and you put it on, and that so that's possible. It's not a problem.
Thank you. And sorry, Peter, perhaps a final question. With a vegan diet, do you recommend this for your patients, for CKD patients? And I know you're not a pediatrician, but would you recommend it also for infants?
Oh, I wouldn't dare to do this. Vegan diet puts a lot of pressure on the ones that do it, because you really need to be very careful to get all the minerals and proteins you need. I would not personally recommend a patient with chronic kidney disease to be on a vegan diet, but a vegetarian diet, that would be no problem.
Yes.
But
Yeah.
And of course, this should be done in close collaboration with dietitians and nutritionists.
Yes, indeed. Absolutely. Absolutely. Yeah. Thank you very, very much for that, Peter. That was a fascinating talk. Really excellent science. I love your out-of-the-box thinking and experiments and studies. And thank you for finding time to talk to us on the VitaFlow webinar. I'm sure you've left lots of people really inspired, really fascinated. And I don't think I'll be walking back into a grocery store the same way again. Thank you very, very much indeed. Thank you.
Bye.
Thank you. I think we have a few, just the few slides from Jane at the end. Thank you, Jane.
Also, thank you very much, Peter and Rukhsana, for your time today. We hope the presentation has been informative and helped advance your knowledge in this area. The next webinar, hosted by VitaFlow, focusing on paediatric kidney disease, will be by the Paediatric Renal Nutrition Task Force on the 28th of February, focusing on nutrition and acute kidney injury. This concludes today's webinar, and on behalf of VitaFlow, we'd like to thank you for tuning in. Please take the time to complete the evaluation questions before you leave the session. Goodbye and enjoy the rest of your evening.